All right, let's go ahead and get started. Are we good? Yeah, we're good. It's Friday, thank goodness. We've got this nice long weekend. I'm sure we're all looking forward to it. So we won't see you guys again until Wednesday. But for today, we're going to finish up talking about cytoskeleton. We talked about microtubules and we started on kinesin. We learned a little bit about how it walked. I want to show you another example of that so that we can really get that down. And then we're going to move on to dynein. Dynein is going to be moving in the opposite direction that kinesin did, but it'll still move on the microtubules. So this is how we're going to be able to move things back and forth on this microtubule. From there, we can start talking about F-actin. That's our other transport cytoskeleton. This is a lot smaller, and it is short distance transport. We'll look at how that's actually arranged in a cell. But this F-actin is going to be a polymer of the globular actin. We call that G-actin. So those are the monomers that form together. Just like microtubules, there will be an endedness to this. There will be a plus end where you can add more G-actin and a minus end where you can take that off. And then it's going to have a car. You know, what's the point of having a highway if you don't have something that can actually move on it? So we'll have another molecular motor ATPase. In this case, it's going to be myosin. Now, we're not going to get into a lot of specifics with myosin, mainly because it's not one molecule. There's like 19 different myosins. Only some of them are actually the molecular motors that we're referring to here, but they differ slightly, so we're not going to really get specific about them. We'll keep that more generic. We may just finish up with enough time left to start into an introduction of nucleic acids. This is what we're going to spend most of next week on. And nucleic acids are really important because that's your genome. That's how you're passing information on to your offspring, but that's also how you're keeping your DNA straight when you're replicating. Question?
I don't think your mic is on.
That one seems to be on. Is this one okay?
I can hear your hand touching it, but I can't hear you out of the mic.
Maybe I just need to talk a little louder. I'm trying not to shout with the microphones, but I can definitely talk louder. I'm good at that. Is it better for you?
Yeah, thank you.
Okay. So this DNA is really important because every time we make new cells, we have to make new DNA as well. And we need to transcribe that faithfully. If we don't, we'll end up with mutations. And those mutations can cause problems. They can cause illness, sickness, even death. But not all mutations are actually bad. Some mutations can give you advantages. They can help you adapt to new environments. So we're going to have a certain amount of mutations, and that's good, but we want to keep it, it's kind of a fine line between keeping it faithful and allowing for that adaptability, that evolution. The nucleic acids, the differences between DNA and RNA structure, it's going to be differences in their nucleotides. That's the monomer for your nucleic acids. Each nucleotide is made of nitrogenous bases, or just bases as we typically call them. For the DNA, those bases are adenine, cysteine, thymine, and guanine. RNA is going to be three of the same, so adenine, cysteine, and guanine, but instead of thymine, you now have uracil. The other major difference is going to be in this 5-carbon sugar. They'll both have a 5-carbon sugar, but one of those, the RNA, where did I write RNA? There we go. That's going to be a ribose sugar, whereas the deoxyribonucleic acid has a deoxyribose sugar. Both will have phosphate groups attached though. And we'll talk about how we can use those phosphate groups to connect those individual nucleotide monomers into a strand of DNA. We're not going to get to that today, but we'll talk about that next Wednesday. So the first thing I want to do is show you a video. I think I told you guys about this video. It's under Tools for Success on the BMS 300 page. I'm not going to put the audio on because I'm not sure. I know it'll come through the classroom. I'm not sure the video will pick it up, so I'll just narrate. But if you watch it yourself, definitely put the audio on because it's got great sound effects. Come on. I broke it. Aw, man. All right, it looks like it's going to take a minute to warm up. I could do an interpretive dance until then. Or we could just all take a nice breath. There we go, and oh, this thing is crazy. Yay! Okay, that's kinesin. What's shown in blue there is the ATPase feet. That's where you're going to bind your ATP and be able to cleave or cut that last phosphate group off and make ADP and PI. You can see the coiled neck, and then you can't see where it's actually holding onto its cargo. You won't see the adaptor proteins in this. I probably should have let it run because apparently it takes a little bit. Yeah. They use terms we don't worry about, tethers and neck linkers. Here we go. So you have your ADP, because it's at rest, it's in cytoplasm. Now the foot's going to come in contact with the microtubule, and when it sticks, the ADP is going to leave. Now you have a free site, ATP can come in. Once it finally gets there, that ATP is going to cause the coil to change and the foot to flip forward. Do I have this running slow? No, I'm just impatient. Okay. There we go. Flips forward, he sticks. The ADP is immediately going to leave when he sticks, and in the back foot now, the ATP gets cleaved to ADP and PI. So now when ATP comes back in, it's ready for that back foot to flip forward. And it just keeps doing this all the way down the microtubule. Lots of fun. Okay. So that's really nice and slow to show you how it's working.
So is this how the cell uses energy, or how does this work?
I'm sorry, what was the question? Who's even asking it? There we are. Is this?
Like the microtubule, sorry, I guess I'm asking, like, what is the function of that process? Like, what does it do for the cell?
So the question is, what's the function of this? What do we care about this little guy moving except that he's totally cute? He's carrying all kinds of things. He could be carrying vesicles that are bringing proteins that you've made in the ER to the Golgi, and then from the Golgi to the transmembrane or to the plasma membrane. He could be carrying things directly from. Okay, so we didn't talk about this. We gave examples of the vesicles primarily, but they can also carry cytoplasmic proteins. When you make them in the cytoplasm, sometimes they're right where they need to be, sometimes not so much. So you actually do need to transport them somewhere else in the cell. So this is where you're getting everything back and forth within your cell. Okay, of course. All right, so microtubules made up of our dynein, I'm sorry, it is late, tubulin monomers. So here's our microtubule. I'm not going to draw in all of these tubulin. I'm just going to draw this first row. So we're going to orient this. We're going to say that because this is where your alpha subunit of this dimer is, that this is the minus end. And because this is where your beta subunit is, this is the plus end. That means that if we were to stick a kinesin on here, that it's going to be moving towards the plus side of this microtubule, carrying its little vesicle. Now, like I said, it's nice that these videos kind of show you in slow motion how everything is actually occurring. But it gives you this sense that kinesin is really just kind of lumbering along, which couldn't be further from the truth. These tubulins are about 8 nanometers long. These are not numbers you need to know. This is just demonstration. We're moving our foot from one beta dimer to the next. So that means that every step that this guy's taking is 8 nanometers. It turns out that these move about 1,000 nanometers per second. Really rough math here. That's like 100 steps a second. I mean, I had to double check that because I'm like, somebody did math wrong. There's no way. How do you move 100 steps a second? They're fast. Kinesin is really important, and so it has priority. It is the fastest of the ATPases that we'll talk about. Yes?
So since there's 13 monomers in the microtubule, a single one, once it goes through 13, does it just go down to like the next one in the row or like in the cylinder or whatever?
So the question is, how are we polymerizing this microtubule? Because we know, I don't want to do that. I want to do this. That we're limited to those 13 dimers that are lined up around the radius. I forgot to count while I was doing that. Hopefully that was 13. So how do we make it bigger? And the answer is that we are doing end to end at this point. So we add them to make this microtubule longer. And when we add, we will always add 13 more on each of the times that we're lengthening it. Yeah?
Are the microtubules pre-assembled for the kinesin to like do its activity on it, or is it being polymerized while it's walking along?
So the question is, are these all set? Are they already existing for the kinesin to come along and walk on it? In some cases, yes. In some cases, these are pretty stable. And I'll show you an example later. In other cases, no, they're changing all the time. I've seen video, and I don't know if it was just an animation or if it actually came from something that they saw, but it was literally being polymerized right in front of kinesin stepping. Like if kinesin was any faster, it would have fallen off. Okay. All right, so that's kinesin. Now we can also have dynein as our second motor. Dynein again is an ATPase, meaning that it's capable of using that ATP and cleaving it to ADP and PI and using that energy to move it. Dynein is minus-ended directed. Dynein's also a very awkward little guy. Okay, he doesn't really look anything like kinesin, totally different structure. He kind of looks like he's on stilts. He's still going to have feet that serve as ATP binding sites. Okay, so that is your ATPase there. That's your enzyme within that foot. It's going to have the legs. It'll have proteins that can then pick up the cargo, the vesicles or the cytoplasmic proteins to move them. But notice one of the biggest differences here is these legs are not intertwined, meaning that we're not going to use the movement of one foot to cause the flipping of another. These legs are rather independent and also very, very awkward. Okay, if you look for video on this, you'll see. In fact, if you find a video that says the strut of a drunken sailor, that's a really good example of dynein. Yes?
When the foot on the dynein or the kinesin is like off of the microtubule, is that when it has ATP, or is that when it has ADP and like phosphate?
So we're not going to talk about the exact mechanism of dynein the way we did kinesin. That was the one example we gave you. This is, I know it's a different mechanism, so I'm not entirely sure how it works. if we were talking about kinesin, the question is, what do we have bound to the feet when it's not connected? And that would always be ADP. I suspect the same is true here because you can't have ATP bound to an ATPase for any length of time. It's an ATPase, it's going to cleave it. So if it's at rest, it should have ADP. Yeah.
So does the ADP and PI allow the foot to become unattached?
So I know it sounds weird, right? Does the ADP allow the foot to become unattached? We said that ADP is sticky and it lets it stick to the microtubule, and then we said that we cleave the ADP and the foot comes up. What's happening is when you cleave, you're causing another conformational change. It fitted a certain way when it had ATP bound. When it now only has ADP, it's going to change enough that it's not going to be as tightly connected, and then that ATP whipping on the front foot will whip that foot around. That didn't answer your question.
Now,
Ask again.
Saying like the ADP plus the PI, like what does that do to the foot? Like does that allow it to unattach, or does it?
Not necessarily. It will cause a change in the shape of the foot that will make it so it's not necessarily attached the same. We don't talk about, or we're not going to ask you about exactly what happens when ATP cleaves to ADP in terms of the shape. Okay, so I like to think of it as the foot starts to come up in preparation for being swung around, but I don't know if it completely disengages from the microtubule or if that change in shape just causes it to kind of like prep for a step. All right. Any other questions? Yes.
So does it like use the ATP as energy, turning it into ADP? Is that why it changes to be able to take that step?
Ask it one more time.
So does the little binding site use the ATP like as energy and then change it into ADP so that it can move like how we do when we move? Or is that?
So the question is about using the ATP for energy. When you cut ATP into ADP, you release a lot of energy from that bond that you cut.
Yeah.
Okay, so the ATP binding in kinesin, probably in dynein, but I'm not sure, in kinesin, that's actually causing a conformational change. It's not the energy from the ATP that causes the foot to whip. Okay, it's when you actually cleave that you release that energy.
So it's more like letting go of a spring then?
I like the way you're thinking about that.
Okay. Cool.
Good.
Kind of helped.
We're not going to get into detailed questions about this. We wanted to give you this example so you could really see how this energy could be used for movement, but we're not going to like ask you to go step by step through ATP's bound, now it's doing this sort of thing. We want you to understand that it's an ATPase, it's cleaving that ATP to ADP, and that the process of all of this is going to allow for that movement. It's going to allow feet to whip forward and always bind to the beta subunits. Okay. All right, where was I? Dining. All right, yes, our little drunken sailor is now going to be moving in the opposite direction. So it moves towards the minus n side. And just to see that within a cell, okay, those are my very sloppy, hastily driven microtubules. When we are closest to the nucleus, that is going to be the minus end. And when we're towards the membrane, that is the plus end. So if we were to look at how these motors were moving, kinesin will move along the microtubule towards the membrane. Dining, on the other hand, will be moving more towards the center of the cell. Okay. We can do local transport, and we do, but these are also really important in terms of the vesicle traffic that we talked about. So moving things out to be secreted, to be exocytosed, or moving things into the cell via the endocytosis. All right. Any questions about microtubules? Yes.
What kinds of things are stuffed into the vesicle on the dining?
So it depends. The question is, what gets stuffed into a vesicle for dining? And that's going to depend a lot on the cell. If it's an immune cell like the macrophage that we showed as an example, then it's probably picking up potential pathogens. On the other hand, if it's an epithelial cell, it really shouldn't be trying to pick up pathogens. That's not its job. But it might be bringing in proteins, growth factors, nutrients that the cell needs, things like that, that it can find in the extracellular space and bring back to its nucleus. Yeah.
In a microtubule, like perpendicular to the nucleus, which ATPase do you use?
Oh, you mean like I drew this and it's right? Yeah. We're going to make this longer and say that's closer to the membrane. Plus. We're never going to get that specific with you. I just drew that badly. Yes?
Do they ever clash together, or is it just one on one microtubule?
So the question is, do they ever clash together? Are you talking about the motors or the cytoskeleton itself?
This is cytoskeleton.
I'm guessing it probably does. I don't know. That's my answer. I don't know. I would guess probably at some point. There is so much of it. It is ubiquitous in the cell. And I'll show you really that pretty picture I promised you. We'll get to it. And you could see how much cytoskeleton we have. I would assume that it probably has to at some point, and it somehow figures it out. Yes?
What happens to the kinesin when it gets to the end?
I love this question. Okay, so we know that kinesin can only move towards the plus side of these microtubules, and dynein can only move towards the minus side. So what happens at the end? I mean, you get your kinesin down here, it can't move any further. You get your dynein back here to the plasma membrane or to the nucleus, and it's done. So I was super excited when a couple years ago I looked this up because I was curious, and I found this video that showed that the opposite one can carry. So like kinesin gets down here, dynein will pick it up and carry it back. And when dynein gets down here, kinesin can pick it up and carry it. It's so cute. They just like fling them over their shoulder with their little vesicle and they're hauling them down. Since then I found out that that's actually not very common. They managed to find a fairly rare event. The more common method is just that they'll drop off. So kinesin gets down here, he falls off, he can diffuse back, start over again. Same thing with dynein.
Is the negative end always closest to the nucleus of a microtubule?
So the question was, is the minus end always closest to the nucleus? Yes. The plus end will always be closer to the plasma membrane. And one thing, because I know I do this, I try not to, but it's a habit, and I know you will too. When we see a negative sign, we think negative. But we try not to use the terms negative and positive here, because these are not actually charged. This is just an indication of where we're adding or subtracting. We'll talk about things that are charged, and we want to like try to keep that separate. So always try to remember to use plus minus, or just a plus and a minus sign. Yeah.
Why do the microtubules always arrange in that fashion?
Why are microtubules always in the 13?
No, why do they arrange minus end towards the nucleus?
You know, someone asked me this last lecture. I didn't have the answer then. I still don't. I didn't have time to look it up. So the question is, why exactly is it that the minus end is closest to the nucleus and the plus end is closest to the membrane? We understand why there's plus and minus, why it's adding and subtracting, but how does that force an orientation? And unfortunately, I don't have the answer. Yeah.
Where do ATPases that are at rest go? Like, is there like a little like parking lot for all of them, or like what do they do?
Yes. They chill at the local bar. They're actually always on a hunt for the new. They're like Uber drivers. They're going around looking for their next fare. So as soon as they can pick up a vesicle, they'll try to find a microtubule. It's just that, I'm anthropomorphizing. They're not actually looking for one. They just sort of randomly move around until they find their vesicle, and then they find that microtubule.
Okay, so you said that at the end the kinesin falls off. They would have to connect to another microtubule. What's the force that allows them to find the other microtubule?
So the question is, when they fall off, how do they find another microtubule? What force is that? I was always taught it's just Brownian random motion, and that they sometimes run into a microtubule, not that they're kind of attracted to one or target to one. Okay. Yes?
Do the feet on dining still bind to the beta, or do they bind like the alpha?
So the question here is, where are the dynein binding to? I've looked that up. I can't find an answer. Like I said, we don't get into the details of this, and quite possibly because there may not be discovered yet. I was not able to find an answer to that. I thought I saw another hand somewhere. Nope. Okay, then let's go ahead and start talking about filamentous actin. And green. So filamentous actin is another transport cytoskeleton. It's much smaller than microtubules, but it is still a polymer. This filamentous actin is polymerized from G-actin or globular actin. This is very well named because it's just a glob. Like so. Probably the easiest thing you have to learn in this lecture. Hey, G-actin. Just like our tubulin with microtubules, these can self-assemble. So you'll just tack a bunch of these together. Okay. So that is your G-actin. This is your polymer, F-actin. So I told you that this had an endedness, just like microtubules. There is a minus end that you can subtract more from, and a plus end that you can add more onto. Here's where it gets a little bit complicated. There's nothing about the G-actin itself that gives it that orientation. Okay, these are just blobs. Yeah.
And you said that those minus and pluses are not charged, correct?
Right, so when we say minus and plus end and we're showing these positive and negatives, these are not charges. There's no charge to this. We're just indicating which side we can add to, plus, or which side we can take from, which is minus. Okay, what was I saying? That there is no orientation to the G-actin itself. What's giving you your orientation is a bunch of proteins that help this assemble. Okay, so unlike microtubules, we actually add a whole bunch of other factors to this polymer that we're not going to talk about. We're not going to get into, we're not going to name, we're just going to pretend they don't exist, except for the fact that you kind of have to know they're there so you understand how we have that endedness.
In this case, does the minus end also stay closest to the nucleus, or does it not matter?
So the question is the orientation here. When we say minus end, is this going to be oriented just like the microtubules, where the minus would be closer to the nucleus? And the answer is yes. So if this is actin, the minus end will be closer towards the center, and the plus end will be closer to the plasma membrane. All right. So now we have our highway. We need a motor. And And we're going to use myosin. Again, there's a lot of different types of myosin. We're not going to differentiate them. We're just going to generically talk about myosin. Some of them do have different structures, but they're all, again, ATPases. So they will all have that ATP binding site to be able to cleave the ATP for energy. Most of them, the ones that we use on actin to be able to transport, are fairly similar to kinesin. They will have those coils. So their feet work together, unlike dynein. So we can kind of draw it similar to kinesin. Now some of these are capable of moving to the minus end. And let me rephrase that because I don't think that was clear. Some of these will only move to the minus end. Different myosins can move only to the plus end. So they always have direction. We'll have, just as an example, we do not expect you to differentiate them, but we'll have, let's see, myosin 6, which is negative, or sorry, minus ended. So he can move this way. But then we'll have myosin 5, which can move towards the plus end.
Can any of them move both directions, or can all of them move one direction?
Can any myosins move in both directions? I'm not 100% certain. Every myosin I know of only moves in one direction, but there could be some that I just don't know.
Okay, but as far as this is concerned.
As far as we're talking about in this class, they will always be one direction only. Yeah. Okay. Okay, so this again is your ATP binding site. This is where you can cleave your ATP to ADP and PI. That's it. That's all I have. We went into a lot of detail for kinesin, but now the rest of them, what we want you to understand is that these motors are always ATPases. We want you to know what direction they move, and we want you to know what cytoskeleton specifically they can walk on. So myosin on actin, dynein on microtubules in the minus end direction, kinesin on microtubules in the plus end direction, right? And again, I'll point out that we had our long distance transportation. So we said that microtubules were long distance. And that actin is short distance. So if you're carrying a vesicle, and I know this comes from the nucleus, but let's pretend this kinesin was actually picking something up from the Golgi apparatus, and it's taking it to the plasma membrane, it's not actually going to get it to the plasma membrane. Okay? It has to transfer its cargo to myosin on actin to get it to the membrane to exocytose it. So here's our little kinesin. I'll actually do it here. He's got to hand off that vesicle. He's got to hand this vesicle off. And that's how you actually get it exocytosed. The microtubules don't go all the way to the membrane. They have to have that actin.
Is there any specific way that you can know which myosin would go which direction?
look it up. Which myosin goes which direction? I don't actually have them all memorized. I know 5 and 6 and 4. We'll talk about that. I don't know the rest of them, so if questions ever come up, I literally Google it. Yeah.
Is there a certain reason why it can't go all the way to the end? Like that they can't cross over?
So why can't the microtubules go all the way to the end? I don't know why exactly. I look at it as a traffic pattern. Like you need to, here's how I always explain microtubules versus actin. I say that the microtubules are like when you fly into Denver. Okay, you're gonna come into a specific terminal and you're gonna have a ton of flights coming into that terminal. But you don't just get off the plane and then walk out to your car from there. You have to go through the terminal and get onto that little train that goes underground with all the fans on the walls. That's gonna take you to the exit where you can actually get out. So microtubules would be your planes coming into the terminals, and then the actin takes you to your actual parking lots. So it's just kind of a way to feed and control the traffic. Yeah.
So is the actin only between the microtubules and plasma membrane, or is it also in like other pieces?
So the question is, we showed the actin is close to the membrane here. Is it always that way? No, it's primarily. We have a lot of actin around our plasma membrane to be able to do this, but we can use actin in other places too. It's not always right there. And of course, not all actin functions like this. We'll talk about actin and myosin again when we talk about skeletal muscle. Yeah.
So I was wondering, does dynein attach to the alpha side of the tubulin monomers? Because I know kinesin only attaches to the beta side.
So the question again is where exactly is dynein connecting on the microtubules? We know kinesin has to walk on the beta subunits. Where is dynein connecting? And unfortunately, I have not been able to find the answer to that. I've looked and looked and I can't find it. So I don't know if it's not known or I'm just not doing a very good job of looking at it.
So actin is part of the cytoskeleton of the cell. Is that what that is?
So the question is actin part of the cytoskeleton? Yes, so is microtubulin part of the cytoskeleton. They're specifically transport cytoskeleton versus structural, like intermediate filaments.
And then so during endocytosis, like a vesicle pops out of the cell. Is there actin inside of that vesicle, or like is that become a part of that structure now?
Okay, so the question is, if you are endocytosing from your cell and you have this actin that's pretty well connected to the plasma membrane, are you going to end up picking up that actin within the vesicle? As far as I know, no. As far as I know, that actin stays there. You're going to build your vesicles around the actin to be able to pick stuff up from the inside, and then the myosin on the actin will grab that vesicle to carry it back to the microtubules to take it where it's going.
Does the actin bind to the microtubule? Because if you're having the ATPase from the microtubule transfer to the actin ATPase, is it like, are the pathways bound at some point, or are they kind of like launching the vesicle from the microtubule to the actin?
So the question is, are the microtubules and the actin actually connected? Or when they're doing this handoff, are they having to throw the vesicle to the next guy? I really like the image of them tossing their vesicles to each other. I don't believe that they're connected in any way, but I imagine that they probably do get fairly close.
So does that mean that like the microtubule in the cytoplasm kind of can wiggle around or wave, or is it a pretty rigid structure?
The fact that the microtubules, because it has that 13 tubulin radius to it, that gives it strength and stability. I would not expect a microtubule to be able to wiggle.
Okay.
Do vesicles, when they're moving back to cells, have centered arteries to tell them which way they need to go, or are they just picked up by any random motor that's around?
So the question is, do these vesicles have any kind of signal on them that targets them? Or are they just randomly picked up by anyone and who knows where they're going to end up? They have to be targeted. They'll have probably sugars, glycosylation patterns that will target them to a specific area. Okay. All right. So I want to show you the cytoskeleton network in a different type of cell. This is just our generic cell. Most cells don't actually look like this, but this is how we kind of diagram a cell for you. But there's a certain type of cell that looks very, very different. And so it has a different type of cytoskeletal layout. And that's going to be the neuron. So I'm not going to get into a lot of detail about the neuron because I'm not going to take Dr. Warren's thunder. But a neuron has a cell body that's basically everything we've talked about with a cell. This is going to have your nucleus. It will have your endoplasmic reticulum, Golgi apparatus, it'll have ribosomes. Basically, it will have all of the protein synthesis machinery that it needs to make proteins here. But now it's got this really long process that we call an axon. And then at the end of the axon, it has an axon terminal. So in cells, when we talked about picking up a vesicle from Golgi apparatus and taking it to the plasma membrane, you could really take it to any part of the plasma membrane you wanted to. You could send it out anywhere. Here it's different. Here you're going to bud this off the Golgi, and it has to go all the way down to the terminal. So if you get this vesicle with its secreted proteins in here, it has to come all the way down here to merge and exocytose. So in this case, we're still going to have our cytoskeleton that's going to be in this cell body, but we now have these microtubules that are going to be running down this axon. Now to be clear, when we're talking about neurons, we could be talking about something relatively short, or we could be talking about something that's going to have a cell body in your brain and then have to go all the way down to your lower spine. It could have a cell body in your lower spine and go all the way out to your toes. So these are really long. So these processes are so long that you can't just have a single microtubule that whole way, then it would no longer be stable. So instead you just have multiple microtubules. And kinesin just hops. These are like bus transfers. So here's our kinesin. He's moving this way because we're oriented such that this is the minus and this is the plus end. Once he gets here, he'll hop down, pick up this kinesin. He'll pick up here and just keep traveling, hopping along until he gets to the end. Now when he gets to the axon terminal, we're going to have actin with myosin. Okay, so again, kinesin will get all the way down here and then have to hand off that vesicle, maybe have to toss it to the myosin to pick it up and take it to the plasma membrane to exocytose it. And again, these are going to be oriented such that that's the plus and that's the minus side. So hopefully that gives you a better visualization of what we mean when we say short distance versus long distance. These are truly long distance versus short distance. There's one other difference with these neurons from the other cells we've talked about. When we're talking about a generic cell, we may be moving some things occasionally from the ER to the Golgi, and then maybe at another time, we're actually moving a whole bunch of cytoplasmic protein somewhere. So we need highways in different places, and we can move them. So we said we repolymerize. We take them off, we put more on, we move them around. Your neurons are stable. Once you've set them, they're going to stay. So if you are looking at these particular cytoskeletons, the microtubules and your actin, they're stable. You don't need to change these. You don't need to move your highways. So we can stabilize these now by adding proteins. We'll essentially cap them. Okay. So any other questions about cytoskeleton or the molecular motors that we're using on them? So I promised you that pretty picture. Here it comes. This is not just a picture, actually, it's interactive. And I'm going to post the link to this under the Tools for Success on the BMS 300 page. It is very detailed. It is well more than you guys will ever need for this class. But there will still be some things that we can recognize. Okay, so this is the interior of a cell. This is an image that was taken by combining things like X-ray technology, electron microscopy, some NMRI. They actually did protein labeling and found every different protein in the cell. And we have individual pictures, I'll show you some, that if you highlight something, it'll tell you what that protein is. So it's a lot of fun to play with. Hopefully you guys recognize this right off the bat. Anyone? Double membrane, what's it tell us? Mitochondria. What about these? Anyone guess what these are? Nuclear pores. Okay, so that's telling you you're at your interface between your nucleus and the cytoplasm. Here's your nucleus. You can actually see some DNA. And then just outside, you can see these ribosomes and the mRNA. This ribosome is attached to a part, excuse me, of the ER. So that's a membrane-bound ribosome that's actually making either plasma secreted or lysosomal. I'm sorry, transmembrane secreted or lysosomal. So I'll let you guys play with this, but I want to just show you one particular thing. Oh, that was actually what I wanted to show you. All of this right here, this is a microtubule. That's a microtubule. All this up here, this is actin. So you can see how ubiquitous it is in the cell. But you can also see, if you pull out, that this is your plasma membrane. So that's the outside of your cell. And you can see how your actin tends to be congregated around that plasma membrane to be able to help get those vesicles close enough to exocytose. All right, so we'll pick up with DNA when I see you guys next Wednesday. Have a nice weekend.